Hello to everyone, my name is Giacomo Baruzzo, I'm a researcher at the University of Padua, and in this talk I'm going to present you our tool scSeqComm, a new tool to infer inter and intracellular signaling from single-cell sequencing data, a tool that is also published recently in bioinformatics. When we talk about cellular communication, we talk about one of the possible ways in which cells can interact each other and trigger a response in other cells. Cellular communication can be split in two main parts, an inter-cellular signaling part, where different cells exchange some messages, often through a ligand-receptor mechanism, so where a protein, the ligand, is secreted by a cell, and it's received by another cell thanks to another protein, called receptor. And an intracellular part, when the receptor that have feeling the incoming intercellular signaling then activate several signaling cascades inside the receiving cell that at the end trigger the response of a cell to the incoming cellular communication. For example, the activation of a transcription factor and the change in the expression level of some genes. Cellular communication have an important role in many biological processes, for example cell development, and diseases, for example cancer. And so it's very important to have robust and reliable bioinformatics method to analyze cellular communication. The advent of single-cell RNA sequencing data provided a new way to study cellular communication. We know that it is possible to computationally identify group or cluster of cells, and then try somehow to quantify the communication molecules and the communication effect, both at RNA level. However, the mechanisms that I showed you in the previous slide, such as the linker-receptor pairing, happened at protein level, so we should be aware that RNA is just a proxy of protein activity. Moreover, the analysis of single-cell data is a well-known challenging task due to the high level of noise present in this data and the difficulty in extracting reliable biological information from them. Even for this reason, the bioinformatics analysis of cellular communication is probably the most recent class of bioinformatics analysis that were developed in the single-cell field. And if you compare them with more established analyses such as cell clustering or trajectory inference, realize that the bioinformatics analysis of cellular communication is still not part of a typical single-cell analysis workflow, and that this class of methodologies is quite recent compared to other methods, and there is a large room for improvement. State-of-the-art bioinformatics methods for cellular communication analysis using single-cell data are mainly focused in inferring only the intercellular signaling part, meaning that current methods are able to provide as output which group of cells are communicating and which ligand-receptor pair were used. Many methods identify which intercellular signals are ongoing, but they do not quantify their strength, and so don't allow results prioritization. Some methods are also able to quantify the evidence of the detected intercellular signaling using different scoring schemes. However, this method often requires the user to set an arbitrary expression threshold to define when a ligand, a receptor, or a ligand-receptor pair could be considered as active, and so they are sensible to the choice of such expression threshold. Even the adopted scoring scheme and the way the intercellular signal is quantified are often hard to interpret, and the prioritization of the results across datasets is not trivial. The second main issue is the lack of intracellular signaling analysis, and so the inability of many methods to quantify the evidence of ongoing intracellular signaling and the functional characterization of the effect of the ongoing cellular communication. We try to overcome some of the previous limitations with the development of scSeqComm, a novel method that combines single-cell data and the available knowledge present in several biological databases to both infer and quantify the evidence of ongoing intercellular signaling and also intracellular signaling, as well as identify the final effect of the ongoing cellular communication. The main novelties of the proposed method are a new intercellular scoring scheme that tries to overcome many of the issues present in current intercellular scoring schemes, for example, the need to define an expression threshold, and a first attempt to identify and quantify intracellular signaling and to provide a functional characterization of the effect of the detected cellular communication. The method requires as input a pre-processed single-cell count data with cells already classified in different clusters, and a ligand-receptor pair databases that the user can provide as input or use just one of the 21 databases already included in the package. The first step of the proposed method is the detection and quantification of ongoing intercellular signaling. First, for each ligand or receptor in a given cell cluster, we compute what we call the ligand score or receptor score, measuring how much the ligand or receptor expression level is high compared to the gene expression level observed by chance in the same cluster, so in a very data-driven way. Once we have assigned to each ligand and each receptor in each cell cluster a score, we use the information of known ligand-receptor pairs to compute what we call the ligand-receptor pair score. The ligand-receptor pair score is computed as the minimum of the ligand score and receptor score, implementing a fuzzy and logical operator, and mimicking the idea that intercellular signaling through a given ligand-receptor pair is ongoing when both the ligand and the receptor are acting. Having designed the intercellular score in this way has several effects on the characteristics of the score. First, users don't need to specify an arbitrary gene expression level threshold. The score is indeed completely data-driven. Second, the score makes no assumption on the scale of the data and the normalization method used to process the data, meaning that it can work with log TPM as well as scrum normalized data. This is very important because it not required to interpret the score in different way depending on the scale and meaning of the data. Indeed, the score has always a continuous and finite range in 0-1 that make it easy to rank and understand. The value of the score is a function obviously of the ligand and receptor expression level, but in its computation we also consider the expression level of other genes, and so even the number of cells in the cluster and the variability of the data in the cluster have a role in defining the value of the score, such that the reliability of the data is somehow considered during the score computation. This gives to the score a quite conservative behavior, as is done by the choice, for example, of the minimum as function to compute the ligand-receptor pair score. About the identification and quantification of evidence of having an ongoing intracellular signaling, we first put what we call an intracellular signaling gene graph. Basically, we want to provide a modeling of the signaling cascade that happens inside a cell, and we use the information from the KEGG and Reactome pathway databases and several transcription factor module databases to build gene graph that connect receptor to transcription factor and their regulated genes. The idea is that receptors are the input node in this gene graph, and that the intracellular signal is propagated from receptor through transcription factor and their downstream genes. This also tells us which transcription factor are somehow reachable by receptors, and so which transcription factor could somehow receive the intracellular signaling triggered by the given receptor. This receptor transcription factor a priori association is what we use as first information to compute the intracellular score. Indeed, we compute for each transcription factor in the gene graph the personalized PageRank score of its node, using the receptor as a seed node. This is a way to measure how much the transcription factor is somehow reachable by a given receptor, and so have a way to quantify the receptor transcription factor association using the topology of the intracellular signaling gene graph. A second step, we plug the dynamic information provided by single-cell RNA-seq data, following the idea that we should see a transcriptomic response in the given transcription factor if it is involved in an ongoing intracellular signaling. For each transcription factor, we use the results of a Fisher test on differential expressed genes controlled by that given receptor transcription factor to define the activity of the transcription factor itself. Last, we combine the static information from graph topology and the dynamic information from single-cell data to compute the final intracellular score associated to each receptor in a given cell cluster. The output of the intercellular and intracellular analysis look like this. For each ligand receptor pair between any two cell cluster, the method provides the evidence of an ongoing intercellular signaling and the corresponding evidence of a related intracellular signaling, triggering some downstream genes, together with the gene graph that were used to connect the given receptor to the downstream genes. This is a very convenient way to summarize the results and give the user the flexibility to inspect, filter, and prioritize the results in a very intuitive way. Moreover, considering the noise of single-cell data and the fact that RNA is just a proxy of protein activity, having the evidence of both intercellular and intracellular signaling provide a more reliable way to study cellular communication. Last, once the user have identified the most relevant receptor looking at the intercellular and intracellular signaling evidence, we provide a functional characterization of the effect of detected cellular communication through a gene ontology enrichment analysis performed on downstream differentially expressed genes. The method can provide a functional characterization of biological processes associated to the detected communication in each cell cluster, as shown on the left, and even similar results stratified by each receptor, as shown on the right, so the list of enriched biological processes in each cell cluster that were identified using the downstream differentially expressed genes associated to a given receptor. The tool provides also several visualizations of the results. For example, it's possible to summarize and visualize the amount of ongoing cellular communication between pairs of clusters, as shown here in the two figures in the form of a core diagram on the left or heat map on the right. Even more advanced visualization options are available, able to capture both the intercellular and intracellular signaling. Here, the diameter of a circle encodes for the intercellular signaling score, while the color encodes for the intracellular signaling score. From left to right, the plot shows how it is possible to visualize the activity of a single ligand receptor pair across all cell clusters, the ongoing inter and intracellular activity between specific pair of clusters, and last, a custom visualization of a selected list of ligand receptor pairs across selected cell clusters. A considerable effort was done even in assessing the performance of the proposed method. First, we compare the result of scSeqComm with independent cellular communication analysis done in a common dataset. Specifically, we use the intercellular signaling analysis performed by Kumar and colleagues on T cell, where Kumar and colleagues identifying a set of ligand receptor pairs involving ingoing and outgoing communication from T cell, in outgoing only communication, and ingoing only communication. When we put this ligand receptor pair in our analysis, we see a considerable agreement between Kumar and our intercellular score. Indeed, ligand receptor pair identifying having a role in both outgoing and ingoing were identified even by our tool, as well as ligand receptor pair involved in only outgoing communication or ingoing communication. We further assess our method even using spatial transcriptomics data, having the assumption that close cells are more likely to communicate with each other compared with distant cells. For this reason, we compute the intercellular and intracellular score on this spatial transcriptomic dataset in both close and distant cells, and compare the value of the score with a one-Whitney U-test. As you can see from the picture, both intercellular on the left and intracellular on the right score are higher in close cell compared to distant cell, also having a significant p-value. Last, we also compare the prediction of our tool with the one of existing tools, namely CellPhoneDB, CellChat, single-cell SignalR, and the method developed by Zhu and colleagues and Skedy and colleagues. Overall, we saw a very good agreement between our method and other method, and specifically, scSeqComm had the lowest number of method-specific results and a general conservative behavior compared to other methods. We are also continuing to work in this field. In particular, we are currently developing a Shiny app that we call CCLength, a very interactive and effective way to filter and visualize the result of cell-cell communication from the communication result that can come from scSeqComm, as well as for all the main cellular communication tools. And if you're curious, we will have also our presentation, still a ISMB ECCB, that you can follow to have further details. We are also working in extending the communication analysis framework to a differential communication analysis framework, meaning we are extending the tool to consider two conditions, even at patient-specific level, if its data are available, to identify in a robust way intercellular or intracellular communication that are altered across the two conditions. Also, we are working on the computational efficiency side, improving the performance of our tool to make it able to handle very large dataset. We are working mixing different programming language R and C++ using ad hoc R packages, and we were able to achieve a full in-memory computation, also having a shared memory parallelism. Something that allows us, for example, to analyze this amount of cell, almost 250k cells, in just 15 minutes using less than 30 GB of RAM. Also, we are working together with other research groups around Europe to have a systematic review of the tools and the resources involved in performing bioinformatics analysis of cell-cell communication. And even for this, if you want to join us, we have a poster during the poster session. Let me conclude my talk giving you the pointer to the article, to the scSeqComm R package, and to the recent developed Shiny app. And also let me acknowledge the other two authors of this work, so Julia and Barbara. And obviously, thank to all of you for your attention.